My name is Amritha Jaishankar. I'm with the Maryland Stem Cell Research Program, and we are focused on identifying and fostering research and innovation in the field of regenerative medicine in the state of Maryland. And as part of our new initiative called the Accelerating Cures Initiative, we're also committed to adding programs to move these discoveries from the bench to the bedside, as well as add mechanisms to build and grow stem cell companies in Maryland. And we have invested about $140 million so far in furthering this field in our region. So I think it's interesting. I think that the concept of organoids per se has existed for a long time, and our interest in understanding how morphogenesis occurs has been a long-standing question. But with the ability to reprogram human somatic cells into induced pluripotent cells, and with all the progress that's been made in understanding these developmental paradigms and in being able to come up with new and improved protocols for in vitro differentiation of cells, this all sort of culminated in the organoid technology as we know it today. So I think one of the biggest challenges that people are still trying to resolve is reproducibility. And I think that while organoids represent a tremendous advancement in the way we model disease now, because it's a three-dimensional structure that can self-organize to recapitulate development and architecture of the tissue and the genetic signature of the tissue, as well as numerous things that static planar 2D cultures don't normally allow us to do. But I think that the biggest hurdle right now is trying to figure out how variable these structures are, how to scale this up, how to have the existing technologies that we use to make real-time assessments and microscopy, how to adapt all of these to a three-dimensional culture system, I think is one of the biggest next challenges. And then also as far as clinical applications go, there's also a need to better understand the matrices and the scaffolds that we use in order to make this more translational. There's also a need to include more cell types and recreate the microenvironment in which these organs normally function. And I think that one of the next challenges would be to add immune cells and understand better how diseases can be modeled. So stem cell-derived organoids have multiple applications, and people are using them for biobanking, for drug testing, and I think this is one of the biggest applications is in drug discovery, but also testing drugs for toxicity, and in personalized medicine. There's also an interest in understanding how the microbiome or host microbes interactions, or even simply just to understand the molecular mechanisms of normal and healthy tissues. So I think it's very interesting, the concept of being able to make a human body on a chip. So organoids are a fantastic way to model disease, but the fact is that organs don't really exist in isolation, and so the efforts that are currently ongoing to mimic the interconnectedness of organs and to connect a number of these organs on a chip type microfluidic and tissue engineering progress that's been made to connect them to have a human body on a chip would really revolutionize drug discovery and personalized medicine. And you can imagine how, if you're able to create all these different organs from one person, you're essentially putting yourself on a chip to be able to test these drugs, and so I think that would be the next big frontier.